This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 751, recorded on May the 4th, be with you, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 64 degrees Fahrenheit, which is 19 Celsius, and it's mostly cloudy. We've had some rain, which is good, and there's a gentle breeze from the northwest.
Here it's 19 C and cloudy. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 78 degrees Fahrenheit, mostly cloudy. We had some rain this morning and for the past few days that kind of catches us up to the seasonal normal, which is good. And we're looking to a forecast of mid-80s for the next few days of sunshine. I'm looking forward to getting out in the garden. This is good.
Today's Tuesday. It is COVID day. And so you can stay. Don't go away. You can stay. And a lot of people objected to our diversion about panhandles last week, to which I say, let us have some fun. Come on, it's 5 minutes. It's 5 minutes.
And one of my friends wrote and said, I loved the panhandle discussion. It was classic TWiV. You cannot please everyone, right? No.
Well, plus these are probably new listeners who are not used to, stump grinders and that kind of stuff. Okay.
Wobbles
and
Someone said,
whatever.
I listen for science. And I said, you find another place where you get more science than TWiV. Let me know. Twice a week, forget it. Give us a break. We're human.
Exactly. That's part of the point.
I think that's why a lot of people like it, right?
Yeah.
Mm-hmm.
All right. Vaccine education town halls continue, correct?
Correct. And we have continued interest in these. asv.org/education, where you can sign up for a free Zoom town hall, get your questions answered. There's no question that you can't ask. Now, being scientists, if they don't know the answers, they will say, now that's something we don't know the answer to. But they know a lot of answers about vaccines. So I think it's definitely worth checking out. asv.org/education. Tell your friends. Check it out.
I went to an ACME the other day. Sign in the front. Free COVID vaccines. Just walk in. Isn't that great? Just walk in. No appointment necessary.
Yep. And they're talking about, Pfizer maybe being available down to 12 years old by the end of the week?
Yeah.
That would be good.
It's all good. Now, of course, in many other parts of the world, it's not so good. We are very fortunate here. I was looking at a map of vaccine coverage today, and not a lot of countries are doing well. It's not good.
Right.
But we have to get going there, right? Okay, today we have a paper for you. But first, a story that is in the news having to do with the Russian-made Sputnik COVID vaccine and its issues in Brazil. And so this has been covered widely, and I have a couple of articles. This is actually one of these articles sent to me by Derek Lowe, who writes in the pipeline for Science. I guess Science Translational Medicine specifically, right?
Looks like.
Yeah. And he has an article on the 28th of April, Brazil rejects the Gamaleya vaccine, and then another article on the same day, Russian vaccine behavior. And he really expresses himself in these articles. And so what happened here, there's the Brazilian drug agency, ANVISA, which is Agência Nacional de Vigilância Sanitária. It's responsible for approval of foods, cosmetics, tobacco, pharmaceuticals, etc. And they said, this was their statement, every single lot of the Adeno5 Gamaleya shot appears to have replication-competent adenovirus in it. So I thought this would be good to go over, because who better to tell you all about adenovirus than someone who worked on it all her career and another DNA virologist. So that'd be Kathy and Rich. So we're going to tell you about the science, and then we'll tell you, we actually have some virology friends in Brazil who have enlightened us. So first, Kathy, explain what is the issue here, please.
Okay, so there's early genes that are encoded by the adenovirus, and early just designates a time point before the viral DNA gets replicated. And the key one for this discussion is early region 1, and maybe specifically early region 1A, although almost all the literature is talking about early region 1 for what we're doing today. And this gene is really important for getting gene expression from all the rest of the viral early genes, which then is important for getting DNA replication and expression of the rest of the late genes. So early region 1 is key to replication for the virus. And so it's been found that if you can delete early region 1 from the viral genome, that the virus will not be able to replicate very well, if at all. But then you ask, well, how can you even grow this virus? Well, what you have to do is make what are called complementing cell lines. And in the case of adenovirus early region 1, both 1A and 1B, a cell was produced in the 70s by Frank Graham working in Alex van der Eb's lab in the Netherlands, where 293 cells is what they're known now. So it was a culture of mouse embryonic kidney cells were transformed with adenovirus, and this resulted in the integration of early region 1 into these human cells, chromosome 19 specifically, and it's just a single length of the early region 1 gene. It's not duplicated, it's not messed up. And that presence of that early region 1 in those cells, which are called 293 cells, allows you to grow a virus that lacks early region 1 because the E1 from the cells complements the mutant. There is the possibility that from those cells there can be recombination, and you could, at some low frequency, get a virus back which now expresses the E1 that it picked up from the complementing cell lines. So this is the fundamental issue here that we're going to talk about. But the situation is that this kind of recombination is pretty rare, and there are standards which these companies that are growing these vaccine vectors have to meet to show that their virus vaccine preps are truly non-replicating. They're not going to be able to replicate on a cell line that doesn't have early region 1. And in fact, there's now 2 cell lines that are used in producing the vaccine vectors. I think for the AstraZeneca and the Sputnik virus, it's 293 cells. And for the J&J adenovirus vector vaccine, they're grown in another cell line called PER-C6. And those are also cells that are expressing early region 1, and they were designed specifically to have very little sequence that could allow recombination with one of these early region 1 deleted vectors. So the PER-C6 cells were recognizing this possibility and maybe a little bit better. But what's done here is the virus preparations are made, and we have this document from the Ministry of Health of the Russian Federation describing how they assayed their vaccine preparations using Plaque assay, which is really terrific. And you can go through and look at the details. And then they present the Plaque assay results from 3 experiments or 3 batches. They have a good positive control sample, and their dilution series is good, showing how many plaques they got. So the Plaque assay itself worked. And then they have all their different, it's actually multiple batches and 3 different pages of those. 0 plaques in the lowest possibility. And so what they can say is that the number of replication-competent adenoviruses per dose is less than 50. So I'll stop there and see if Rich and Vincent want to insert additional information I might have missed.
Let me see. I just want to backfill a couple of things. First of all, I would refer people who might be struggling with the concept of the different vaccines and viral vectored vaccines. Refer them again to that YouTube video that I made that describes how viruses work and how the vaccines work and how immunity works, and that should serve as reasonable background. To make sure that people understand that the deal here is that this is a virus vectored vaccine, that the Gamaleya vaccine is in this respect similar to the AstraZeneca vaccine, similar to the J&J vaccine. All use adenoviruses that are different serotypes. The AstraZeneca from chimps, the J&J, a human adenovirus, but a different serotype. The Gamaleya vaccine, 2 different serotypes of adenovirus, adenovirus 5 and adenovirus 26. And what's been done in these is to splice in the gene for the coronavirus spike protein, and therefore use the viral, the virus and the viral genome as a vector. Viruses are very efficient at getting genes into cells, okay? So if you grow up this thing with the coronavirus spike gene in it, you have a very efficient delivery vehicle to get it into cells. Now, there have been developed, not in the market for SARS-CoV-2, but in general, in the realm of virus vectored vaccines, both replication-competent and replication-incompetent viruses. And so the notion, and there are arguments for and against using either one. So the notion that you have a replication-competent vector is not necessarily damning. And these are human adenoviruses that people encounter all the time. So even if there were replication-competent virus in one of these vaccines, it doesn't mean it's going to kill you or something. It probably won't. Okay, but if you're marketing a replication-incompetent vaccine and you have safety protocols that show that it contains no replication-competent virus, you better stick to your guns. Okay, that's the expectation.
Right. And it could be that if there were significant amounts of contaminating replication-competent virus, that it could be a vaccine that you wouldn't want to give to immunocompromised people. So adenoviruses are not really very serious, except for the immunocompromised.
I have another wild-ass subtlety here, okay? And that is that now I don't know about the Gamaleya vaccine, but for most of these, and I'm sure for the J&J vaccine, the spike protein gene is actually cloned into the position where the E1 gene is deleted. So if you recombine back into that, the E1 gene, chances are you'll lose the transgene. Okay. Now it's hard to imagine a situation where an entire batch of vaccine all of the viruses would have been, rearranged to be having E1 and no spike. But that is a subtle consequence of replacing back in the E1 gene. Also, and we'll probably get to this, there are theoretically other ways to get what appears to be replication-competent virus out of an E1 deleted virus. I think, Kathy, we talked about this some time ago. You reminded me that if you have a large enough dose of the E1 deleted adenovirus, you can get leaky transcription, they call it, okay? Just a little bit of gene expression. Now, I have a hard time believing that would give you plaques, because plaques start with a single virus particle, but it's an issue that comes up.
Right. Can I ask, this document that we have here from, it's from Russia, Russian Federation. It's a response to ANVISA, right? Mm-hmm. Is this a public document, do we know? Or is this a private thing?
I don't know.
It says to whom it may concern.
And this we got, okay. So it's very detailed. And the point here is that you do a plaque assay on cells that do not provide E1, right? And what kind of cells would they be? Do you know offhand?
I think A549 cells.
Okay.
Yes, that was it. It was A549.
That's a lung
That's a
cell.
human lung cell, yeah.
All right. So I'm looking at the plaque assay results here, and they have a lot of batches where the lowest dilution they do is 10 to the minus 2. Why don't they use undiluted vaccine and get the less than, even lower than 50 per dose? Any insight there? I would put undiluted, wouldn't you? You guys probably.
Well, so I don't know. My own, when I did stuff like this, 10 to the minus 2 was always my lowest dose. And there were two reasons for it. One was that when I wanted to assay something, I didn't want to use up all of my sample in the assay.
Sure.
Okay. So I would take a small portion and do a dilution. And I knew that 10 to the minus 2 was, I didn't really need to go lower than that. The other reason that comes up sometimes, I don't know if it's relevant to this, is that there may be other things in the preparation. So that if you throw it on undiluted, it kills the monolayer, and you can't tell whether there's any plaques.
Like fiber, right? Could kill the cell, right?
Mm-hmm.
Yeah.
Or, maybe if you have a huge dose of replication-incompetent virus, I don't know, maybe you'd know this, Kathy, would that kill the cells?
Well, I think that's what Vincent was alluding to. If there's just a lot of fiber, which is a toxic protein in itself from the virus. So it's possible that if you have a lot of non-plaque-forming virus, it could be.
Yeah, I suspect that if you use anything more concentrated, okay, that it kills. And people need to understand that the way you see plaques is that these are foci of infection on an otherwise healthy monolayer of cells. Okay, so you have to have a background of healthy cells, and if you throw something toxic into the plaque assay that kills those cells, then you can't see the plaques because you don't have that healthy background.
Okay, and
my guess is that it's just a guess.
Yeah.
But that anything more concentrated than 100-fold dilution of the sample has that kind of toxicity in it that makes it impossible to read the plaque assays.
So this is now expressed less than 50 adenovirus per dose. I presume that's the dose that would be injected into.
That's my presumption as well, yeah.
So what do you think? Is it possible that there is some, or is it just that's the limit of the detection, so they have to say that?
Personally, I think that, from a strictly scientific point of view, the accurate statement is that it is less than a certain amount, and you can't say anything beyond that.
Yeah.
I would. I can't tell in their manufacturing protocols, but I would hope that there were other tests as well. I mean, PCR, for example, on this, if you had a rearranged region, you might be able to detect that. At what level? I don't know.
Yeah, you should be able to do PCR for E1, right, and see if it's in the genome.
But it could be that, in fact, the plaque assay may be, in fact, the most sensitive assay for whether there's actually a problem. My opinion is, I doubt very much.
Yeah.
If there's any replicating virus in this, there's certainly no evidence for it. There's no precedent for it. But I think that they've described their results properly.
Also, I think that, if it happened, it would be in one batch. They're not making subsequent batches, from one where a replicant-competent virus would have arisen.
Yeah.
So, which would amplify it, and you would see it eventually. So, but I agree with Rich. You can't prove a negative, and that's why they have to state it the way they do, which is that they can state that there's less than 50 per dose.
And it would be interesting to see what sort of quality control protocols the other ad vector manufacturers have.
Right.
But I have a hard time believing that they're all that much more stringent than this.
Right.
We should also point out that this is actually there are 30 issues that were raised by ANVISA, and they're all answered here. Besides this replication competence, right? That was number one. But then there's Russian side did not provide personal access for inspectors. They did not get data on sterilizing. They didn't, on and on.
Yeah, and, to the one about, you didn't show us all 7 facilities. We showed you the ones where you're going to where Brazil is going to be getting their vaccine from, and you didn't ask to see the other ones, is what the Russians say. So they have kind of an answer to almost all of them.
And I find that this formal correspondence, at least back from the Russians, I didn't see what ANVISA actually sent them, except that I guess these issues in the document are quoted from ANVISA. But this strikes me as a perfectly straightforward exchange of, we have these questions, here are your answers. In contrast to sort of political brouhaha that you see in the press, with people getting all puffed up and accusing each other of stuff, which is, unfortunately what the press sees. The real back and forth is quite professional, put it that way, seems to me.
Yeah, this article by Derek Lowe, he writes, the news that Sputnik contains replicating adenovirus was surprising and unwelcome. That's not correct, right? That's a misinterpretation of the findings. I guess he didn't have this document. But then he goes on to say, the response on Twitter has been horrible, right? The official Twitter account has accused ANVISA of inventing fake news. And then the official statement of Sputnik camp is no more conciliatory. He writes, a tip for the vaccines manufacturers: don't immediately start accusing your critics of bad faith. Step up and act like responsible drug developers. Throwing fits on Twitter is not the answer.
Go ahead, Kathy.
I was going to say, and also that at least one of these Russian Twitter accounts is just really bashing other vaccines, and saying how much better their vaccine is than Pfizer, and other irrelevancies and inaccuracies. Yeah.
But my understanding, correct me if I've got this wrong, but from the way this was written, my understanding is that that Twitter account is not the vaccine manufacturer or the scientists. It is a Russian mouthpiece representing them. Okay. And so I think that allows for sort of this unprofessional attitude to creep into it.
Right.
To me, the crux of this whole thing is this quote, and I'd like to know if this is actually a quote from ANVISA that you read early on at the top, Vincent, that every single lot of Ad5 Gamaleya that they have data on appears to still have replication-competent virus in it. Okay, and that's, as we've already said, that is sort of standing the results on their head.
Yeah.
It's saying that if you say it's less than 50 infectious units per dose, the implication is that there is some virus in it, and that's not implied at all. Okay. Right. So it's a misinterpretation of that.
So I got that quote from Derek's article, who says that's a quote from ANVISA. They said, according to the data we got, and that's the data in this sheet, which we've just discussed, they said it's got virus, but it's a misinterpretation of the data. Yeah.
Early on in this, there was also the early reports left the impression that ANVISA or somebody in Brazil had actually done their own assays on this stuff and found infectious virus in it. But that's not the case. Okay. They're just, there were no assays on this stuff done in Brazil. at least not associated with this flap at this point. They're just reporting on the data that they have from Gamaleya.
Right. And now might be the time to go ahead and quote Eurico, who's our colleague in Brazil. He's been on TWiV. You can go back and check 317, Eurico Arruda. And he says, I think that ANVISA mistook the expression less than 50 per dose to mean that there was replication-competent adenovirus in the vaccine. Less than 50 per dose, to me, was what was the Russian reference value of what would be tolerable, probably due to some leaky transcription. Or it's not even necessarily what would be tolerable, but as we just described, it's sort of the lower limit of detection in their plaque assay system. So, yeah.
So I just want to put this in a little context. So years ago, when I remember one of the first papers we published for some plaque assays, I put 0, right? And the reviewers said, you can't say 0. It has to be less than 1, because even if you use undiluted virus, we take 0.1 ml, right? And so if there were 10 PFU in a mil, you should see a PFU on some of your plates. But if there's less than 1, you wouldn't see any, but there might still be, 1 every now and then. So you have to say less than 1. And that's why you have to say, and they're 10 to the minus 3 and et cetera, the dose volume of the vaccine, that's where they got less than 50.
I used to do this stuff, this kind of reporting almost every day, because one of the things so I worked with temperature sensitive mutants. And one of the things that I'm interested in is reversions of the temperature sensitive mutation. Okay. And the way you assay those is to take the virus and dilute it out and plate it and put it at the non-permissive temperature and see if you can see plaques. But there are so few revertants relative to the rest of the virus, and the rest of the virus is cytotoxic, it'll kill the cells, that there's the higher concentrations of virus kill all the plaque assays. And you have to dilute it out before you have enough of the monolayer surviving so that you can see the revertants. And that gets reported as less than a certain number. Okay?
Right.
There's another article here that's another science article. That's this was from 2 days after Derek Lowe's article. It says, is Russia's COVID-19 vaccine safe? Brazil's veto of Sputnik V sparks lawsuit threat and confusion, by Sophia Martino and Meredith Wadman. And I found this a really nice summary. Okay. They kind of went through the whole thing from top to bottom, much of the same things that we've described, but if that'll show up in the show notes as well. Their concluding paragraph is unnecessary turmoil. I have to say, have been pleased that this didn't boil over more vigorously in the regular press.
Yeah.
Yeah, it boiled over in Twitter. Okay. But it seems to me that maybe the usual science journalists from like the Times and the Washington Post, where I get most of my information, and the PBS NewsHour have kind of stayed away from this. They did not wade into this whole it's contaminated with live virus thing. So I'm hoping that what that means is that they're doing their homework.
Our other friend in Brazil, Clarissa Damaso, who's written in before many times, says, what makes me really angry is that we could be in a much better position. Our national immunization program was created in the 70s and provides free vaccines against more than 15 diseases to everyone, has an excellent structure to distribute vaccines to all regions of Brazil and immunize up to 10 million people a day if necessary. Since 2015, the P&I has been suffering, and the present administration is really willing to destroy it completely. And she sent pictures of vaccinators immunizing poor people in the Amazon and the Northeast. So they need this vaccine, and they obviously have the infrastructure to distribute it. They have immunized a lot of people, but they have so many such a big population that it's not enough. So this is unfortunate.
We've talked about this before. The Brazilian vaccination program is historically remarkable. Okay, they invented national vaccine days, and so they have a really strong record of this. But they've had some issues with COVID.
The bottom line here is that if you have a problem, you do it civilly. You write a letter to the manufacturer and you say, we have these issues. You don't reject it outright, especially if you don't know what's going on. You don't go to Twitter and make a big deal out of it.
Right. I kind of look at this as that ANVISA is sort of being like an FDA examining group and saying, here's the issues that we have. And now the Russian group has come back and answered them. But in the meantime, this idea that there's replication-competent adenovirus has gotten blown all out of proportion, probably misinterpreted, maybe even by the ANVISA people, but they certainly wanted more information about it and several other things. So,
but we don't know the current status of this, right? We don't know if they've decided.
Yeah,
ANVISA's decided to okay it or not. Okay, I hope they do.
And according to Enrico, there have been issues with ANVISA taking its time to get these approvals in the face of these emergencies. So there have been even legal pressure from the Supreme Court put on ANVISA to get their act together. But we'll see. We'll see how that plays out. I hope the global distribution of vaccine is an issue.
Right.
Okay. And
big
this
time.
is just one piece of a very complicated puzzle.
Yep. Big time.
And I also want to footnote, every time I think about Russian science, I think about the fact that for the past 20 years, ever since the shuttle program was done, we've been completely reliant on the Russian space program to keep the International Space Station alive. They have shot up all of the astronauts and all of the cargo in a flawless record of performance. Okay, so just as an example, these are scientists, and there's they've no reason to assume that there's anything other than good science going on.
So Derek Lowe writes at the end of one of his articles, he says, I fully expect to be trashed by defenders of the current Russian government. I've had it happen before. He says, come at me. I have a great deal of sympathy for the Russian people who over the years have managed to make great contributions to humanity while cynically being abused by their leaders who have too often been a series of despots and thieves.
Yeah, I was looking for where that quote came from because I was going to paraphrase it, but I'm glad you found it.
He's got it. He's right. Russian people can do great things. The leaders have been, we've had problematic leaders also.
Yeah, I was going to say, we never have that problem, right?
Right. Our leaders always listen to the scientists.
Oh,
yeah. No, we are not throwing stones. Believe me, we recognize the problem. But as Rich said, they can do very good science. I mean, Sabin brought his polio vaccine there, and it was given a clinical trial in 130 million people and got licensed here as a consequence. So no problem there. Okay, I hope that clarifies it for you. We have a paper now, which is Nature Microbiology. The furin cleavage site in the SARS-CoV-2 spike protein is required for transmission in ferrets. I think this furin cleavage site is quite interesting. We've talked about it a bit. This comes from Imperial College London, King's College London, and University of Bristol. We have 5 co-first authors, Thomas Peacock, Daniel Goldhill, Ji Zhu, Laurie Bayon, and Rebecca Fries, and this is from the lab of Wendy Barkley, who was on TWiV in Dublin a long time ago. SARS-CoV-2 spike, remember, is cleaved in 2 different places by different proteases. And the cleavage is needed to expose the fusion peptides, so the fusion of the viral and cell membranes can happen. And there are a couple of different places in the cell where this cleavage can happen. In coronaviruses in general, it can happen while the virus particles are moving out of the cell. It can happen at the cell surface by proteases like TMPRSS, TMPRSS2, which we've talked about before, or by cathepsins in the endosome during entry.
So when you said that it can be cleaved in two places, that can be taken two ways. So the molecule itself has two cleavage sites, and there are physically within the realm of the cell biology of the virus, two places where the cleavages can happen.
And the consequences for the two are different. The first, one of the cleavages, typically the first in sequence, if it happens at all, is between the S1 and S2 boundary, and that's the furin site that we will be talking about.
Right.
And that cleavage can ultimately physically separate the receptor binding domain from the rest of the membrane-bound portion of the molecule. The second cleavage is downstream from that, and it's what's called S2 prime. Is that right? And that's the cleavage that is right next to the fusion peptide and is required for fusion because it's required to expose the fusion peptide. So my understanding is that first cleavage by itself is not sufficient to activate cleavage.
That's right.
And the second cleavage is absolutely required to activate cleavage. And this gets
that's right.
hopelessly confusing.
So for students of Vincent's and mine and anyone else's who have pictures of how the hemagglutinin of influenza has to reveal its fusion peptide, or any of those envelope viruses have to reveal their fusion peptide, it's this second cleavage at the S2 prime site that's necessary to do that for SARS-CoV-2.
So
Vincent, we probably ought to put your little diagram of the protein in the show notes, or make it the show image or something like that, so that when people listen to this, they can actually look at it.
It's actually Jason's from his cell paper last year where they solved the structure. I found it very useful. I cut out all the peripheral stuff.
It's burned into my head.
So we've talked about before, if the cleavage of can occur at the cell surface by TMPRSS, then fusion can occur there, and that bypasses an inhibitor like hydroxychloroquine, which inhibits the cathepsins, the other proteases that can cleave the spike in the endosome. So in the right cell, in the presence of hydroxychloroquine, the virus simply fuses at the cell surface and bypasses hydroxychloroquine. And that's why it didn't work, because in the cell lines, the Viro cells where it was originally tested, you can inhibit you can inhibit the cathepsins, and there's no cell surface TMPRSS for the virus to fuse, so you get really nice virus inhibition. But in lung cells, as we'll see again here, the virus can fuse at the cell surface because those cells have TMPRSS too.
So this is going to be redundant of what you have said, Vincent, but it took me through into the discussion, and to get this home, and I suppose I should have understood it from previous discussions, but basically there's two ways for the virus to complete its entry. One is to get, and fusion is critical to this. You have to do fusion of the viral membrane with some sort of cellular membrane in order to ultimately dump the genome into the cells, okay? That can happen either at the plasma membrane on the cell surface, or the virus can be taken up into an endosome, and it can happen in the endosome, Okay.
Right.
And so we're talking really about, among other things, about the competition between those two pathways. And that is determined to some extent by, or to a large extent, by whether or not the cleavage can happen at the cell surface. If you can cleave sufficiently to expose a fusion protein at the cell surface, then you can get in by fusion with the plasma membrane. If you can't, then the only way in is to be taken up by an endosome, and then you use a different protease, okay, to do the cleavage to expose the fusion peptide and exit the endosome.
And what we're talking about here is the S2' cleavage to expose the fusion peptide, whether that happens at the cell surface or in an endosome. The furin cleavage site is S1S2, and that's the topic of this. What's that doing? Now,
right.
What previously, there have been several analyses of this furin cleavage site. So SARS-1 doesn't have a furin cleavage site. It's cleaved by temperus-like proteases or cathepsins. MERS coronavirus does have a furin cleavage site. And by the way, what is a furin cleavage site? It's a series of basic amino acids, and they're cleaved by cell proteases called furins. For SARS-CoV-2, it's PRRAR, proline, arginine, arginine, alanine, arginine. And they can be slightly different. We talked very briefly about a paper from Vineet Menachery's lab, which was published back in January. Loss of furin cleavage site attenuates SARS-CoV-2 pathogenesis. So he removed the furin cleavage site. In viro cells, it doesn't matter if you remove it. Actually, it reproduces quite well. But in respiratory cells, human respiratory cells, removing the furin cleavage site slows down reproduction, and it reduces disease in hamsters and in transgenic mouse models. All right. So this is telling us that this does have an important role. So the virus will still reproduce without a furin cleavage site, but depending on the cell line, it can be more or less efficient at that. This paper takes it a little further. Actually, there's an interesting twist, as you'll see. So what they do first is to modify the furin cleavage site in a variety of ways by introducing amino acid changes. They actually do one where they put the influenza virus furin cleavage site. This is from the influenza H5N1 hemagglutinin, has a very nice furin cleavage site, and they delete it as well. And then they make both pseudotype viruses and authentic SARS-CoV-2 viruses with some of these changes. And one of the first experiments they do, which is quite nice, is that they say it's been known that, these viruses, when they infect cells, can form syncytia, right? Remember that episode a couple of weeks ago? They can cause fusion of cells by virtue of the fusion activity of the spike. And they say the furin cleavage seems to correlate with syncytium formation. So what they do is produce just the spike proteins in cells, and they measure syncytium formation. The wild type SARS-CoV-2 spike gives you a certain amount of syncytia formation. But if you take out the furin cleavage site, there's no more syncytia produced, which tells you that furin cleavage is probably needed for the other cleavage to give you fusion, or at least make it more efficient. They also look at cleavage efficiency by running extracts of cells and looking for the spike protein. And you can see the uncleaved protein and then cleavage product using an antibody to the protein. It's a very clean assay to see uncleaved and cleaved spike. And many people always ask me, what do you mean by cleaved? It's cut. Think of a scissors cutting the protein. It's interesting that we use cleavage all the time, but not everyone knows what we're talking about. Cleavage has other meanings in the English language. So it turns out that the SARS-CoV-2 furin cleavage site isn't very good. They call it suboptimal. It's not as good as the flu one. The flu one gets very well cleaved, and the SARS-CoV-2 one does not. Anyway, so let's do some entry experiments now. This is very interesting, a little confusing because they're using different cell lines, and even I get mixed up when I read it. So maybe I'll mix you up, but
I struggled with this, and I'm still struggling. Even as we're talking about it, I'm struggling. Okay.
So first of all, they do a very interesting experiment where they mix wild-type SARS-CoV-2 with the mutant lacking the furin cleavage site. They do 70% mutant and 30% wild-type. And just think of it as competition assay, right? They're going to see who wins. And they're giving the mutant a head start. They're 70% of the virus in the tube is the deletion mutant, and 30% wild-type. And then they infect Vero cells, your kidney cells, and then human intestinal cells, CACO, right? Carcinoma of the colon, that's what CACO stands for. And then CALU, carcinoma of the lung, these are human cells. And they also have primary human airway epithelial cultures, where you get cells from the airway epithelium, you can put them in culture. All right, so they put this mixture in all of these. And then they look over time, who wins? By 72 hours, the mutant outcompetes the wild-type virus in Vero cells. In the kidney cells, the mutant wins. But in mucosal epithelial cells, the wild-type wins. Like the HAE, the human airway epithelial cells, the wild-type wins over the mutant. Same thing with the CALU cells. Wild-type wins. But if you take out the cleavage site, can't infect CALU cells. So depending on the cell, again, the kidney cell, the mutant wins. In the airway cells, the wild type wins. And if you take out the cleavage site and just try and infect CALU cells, lung epithelial cells, won't infect it. So furin cleavage site is needed in the lung, which is an important place for this virus, right? But not so much in the kidney. All right, so that's just reproduction. Then what about entry? So they can look at entry of these viruses into various cells. Again, they have cell lines and primary cells. They have 293 cells with ACE2. They have CACO and CALU cells. And they're using pseudotypes as well. A negative correlation between efficiency of cleavage of the spike and entry into 293 cells with ACE. So in other words, the viruses with the wild-type spike actually enter these 293 cells, which I think are kidney, right, Kathy? The 293s are...
Oh, well...
You're not sure?
They were originally isolated from kidney culture, but they seem to have more neuronal characteristics, according to Frank Graham. But then people have done now transcriptomics, and they think that they might be adrenal. So to call them HEC cells drives me crazy.
But they're not.
I just call them 293 cells. They're probably not really embryonic kidney cells. They're not a model for kidney cells.
They are cultured human cells of. They are cultured cells of human origin.
Right. Right.
Gosh, isn't it amazing that you make a cell in the 70s and then people are studying it, doing transcriptomics to find out where it came from for years? Oh, that's so weird. All right, so where was I? The wild type enters these 293s less efficiently than SARS-CoV, which is one of the controls they use. While the mutants that have the furin alterations are better at entering these 293 cells. And if you actually put the flu furin cleavage site into these pseudotypes, it decreases entry in these cells. So in other words, the more efficient the cleavage, the less efficient at infecting 293 cells. In the respiratory cell lines, the opposite. More efficient cleavage, better entry into those cells, which is more or less what we saw with the reproduction assays. More efficient cleavage, better reproduction. Okay? So it looks like in the lung epithelium and in the gut epithelium too, there are epithelial cells, you need better cleavage of the furin site, but not in the kidney or whatever 293 is. You don't need it there either. Okay? You okay, Rich?
I'm hanging in there.
right.
I'm trying to think about a big factor here, right? Is whether or not these cells have cell surface expression
Yes.
of temperus.
Which
is
we'll
that
look
correct?
at. That's right. We're going to look at that. In
Okay.
fact.
So we're coming to that.
We're going to emphasize that, right? And in fact, so now we're going to look at some inhibitors, which will address that question, right? We have two inhibitors. We have camostat, which inhibits temperus-2. That's the plasma membrane protease. And E64D, which inhibits cathepsins in the endosome, okay? In 293 cells, if you treat with camostat, no inhibition of pseudovirus entry. So in 293 kidney cells, we don't care if you inhibit temperus. You know, in Vero cells, there isn't even any temperus, so this is probably why it doesn't inhibit 293 in 293s. But in the Keiko cells, the epithelial cells, camostat will inhibit viruses with furin cleavage sites. But if you don't have a furin cleavage site, it doesn't matter. In KLU3s, which are the lung epithelial cells, the camostat, that's the temperus-2 inhibitor, inhibits entry of all coronavirus pseudoviruses that they tested. So if you are in the lung cell, camostat inhibits, but not in the kidney cell. And the kidney cells basically don't have camostat, so that's not even an option. And if you take out the furin cleavage site, now you have no chance of entering. Another experiment they did, well, let me just summarize this. In airway epithelium, cleavage by serine proteases like temperus-2 is required for entry. That's the bottom line in that. If you take out the cleavage site, you can't get into those cells. Now, what about these proteases? Are they present? So they looked at the production of these proteases. These the three human cell lines that they looked at, the 293, the KLU, and the Keiko, and the primary airway epithelial cultures, they all have ACE2 and they all have cathepsin-2, which is the endosomal protease. But the 293 cells don't have temperus, all right, at the cell surface. They don't have it anywhere, but at the cell surface. And that's why camostat didn't do anything, because there's nothing to be inhibited. And as I said before, Vero cells don't have any temperus either. And they do a series of experiments where they put temperus into some of these cells, right? So they can put temperus into 293s. And you can see that it will promote entry of many of their pseudoviruses, and particularly those with furin cleavage sites. So production of temperus-2 favors entry into cells of pseudoviruses with furin cleavage site. So just remember, the furin site is S1, S2. Temperus is cleaving S2 prime. It's a different site. So the two are working together in some way, okay? Because you put temperus in, you're getting cleavage of S2, but it also is enhanced by cleavage of S1, S2.
Yeah, so my impression was, correct me if I'm wrong, that temperus can do both of these cleavages.
Temperus does S2 prime.
Does S2 prime. Okay. So, so in cases where you have a furin cleavage site present, actually, there's a significant amount of that cleavage that has already taken place during production of the virions that you're using to infect these cells.
That's right.
Correct?
That's right. That's right.
And am I correct that that furin cleavage is it fair to say that that's favors or is even a prerequisite for the temporous catalyzed cleavage at S2 prime. That if you don't have the furin cleavage, you can't do the temporous catalyzed cleavage at the surface.
Yeah, that would be the implication. It's less efficient, right? And that's
Right.
why the
less
entry,
efficient.
That's why the entry is not happening, right?
Right.
Right. So we looked at that. Let's go back and review that. Even in cells with temporess, even Caylus cells which have temporess, if you have no furin cleavage site, you can't get into them. So yes, you're right. The furin cleavage seems to make the temporous cleavage more efficient. Yep.
Right.
Must be some kind of well, as you say, the S1 falls off, and maybe that makes it more accessible to the temporous cleavage, right? All right. Then here's the interesting twist. Furin, the presence of a furin site in SARS-CoV-2 allows surface entry in respiratory and mucosal and intestinal mucosal epithelial cells. And they say other people have proposed that this may be a way of avoiding proteins in the endosome that can inhibit virus reproduction. So there are a series of proteins called IFITs, IFITM proteins, interferon-inducible transmembrane proteins. These are proteins both in the plasma membrane and in the endosome that confer resistance to viruses. How they do it, not quite clear, but they seem to prevent fusion. They seem to make the membrane more rigid. And so if these proteins are present, they will inhibit SARS-CoV-2 reproduction, but they're in the endosome. So the idea is that if you fuse at the plasma membrane, you're going to avoid these. I mean, this sounds great. This is a perfect human interpretation of what's going on. Sounds great
like
to
a
us.
virus.
But who knows if that's actually what. Anyway, so they said, let's look at this. So they're saying, so the furin then may allow avoidance of IFIT proteins. So they have a drug. It's actually an antifungal, amphotericin B. Gosh, we used to use this in our cells all the time when they got fungal contaminants. Do you guys use that at all?
On this recent trip I took, I helped my skipper with a fungal infection in his eye by being the nurse to drop amphotericin B into his eyeballs.
Wow. Okay. we used to use it in our cells when we got fungal infection. Anyway, it turns out that it inhibits IFITM antiviral activity. All right. So IFITM will block coronavirus infection, but amphotericin B will, how should I say, reverse the block, prevent the block, antagonize IFITM. Okay. Take your pick. So they can add this to cells. In 293 cells with ACE2, they find entry of all the pseudoviruses they have is improved by treatment with amphotericin B, showing at least in those cells the entry is through the endosome. All right, in Keiko and Caylus cells, if you have a furin cleavage site change, you get boosting by amphotericin B. But if you have furin, there's no effect. Makes sense. With furin, you enter at the cell surface. Without furin, you enter in the endosome, and therefore IFIT's there waiting for you, and amphotericin can block that. To make that clear? Does it make sense?
Yes. So there's this is now crystallizing for me again. I'll lose it if we're.
Well, you'll lose it next week because you're not going to talk about it outside of today, and I'll do the same thing.
But so the ability to do the S1, S2 furin cleavage helps you to do the cell surface entry, so you don't have to deal with the IFITs through the endosome. Okay, if you don't have that, then you got to run the gauntlet through the endosome.
Yes.
Okay, and so there's a selective advantage, if you like, in having a furin site. Okay, because you don't have to deal with the IFITs.
Yeah. If you're in a respiratory, or if you're in some kind of mucosal epithelial cell, which is where the temporess seems to be.
Where you have, where the temporess is. Yes.
But
and just a little foreshadowing, or maybe we're going to get right to it, but You don't want to have a furin site that's too good.
Yeah.
This is what kind of threw Rich and me at the beginning. If we had read the discussion first and then read the paper, I think it would have helped us. But
the next experiment, they kind of put all this together. They produce ACE and Tempress with or without IFIT in 293 cells. All right. And then they can ask the relative contributions of all of this. So with the furin cleavage site, you have less inhibition by IFITM than spikes that can't be furin cleaved. And that makes sense because that's what we've just showed you already. But here they've put it together by producing the proteins individually. And then the last experiment before we get to the animals, what does amphotericin B do to SARS-CoV-2 reproduction in KLU and human airway epithelial cells? Wild-type virus, no effect, because that virus is getting in at the cell surface. The cleavage site mutant lacking the furin cleavage site boosts. It doesn't grow well in those cells, if you remember, but with amphotericin B, it boosts the ability to replicate.
Right. So all makes sense. Good.
So
We're
it just thinks to me, if you're on systemic amphotericin B and then you get infected with SARS-CoV-2, furin cleavage site mutants could arise in you, right? That's inside baseball, I guess. Right? Because you allow, without furin, you could get in via the endosome as long as there's no IFIT, and the amphotericin B. All right, never mind.
yeah.
So back to Kathy's point, there's sort of an optimum efficiency of this furin cleavage because the cleavage, the S1, S2 cleavage can happen, frequently does happen during production of the virions.
Yeah.
So you got mature virions before they're even going to infect the cell. That cleavage has happened, but non-covalent interactions are holding the S1 and S2 subunits together, and you need that whole intact structure in order to do the receptor binding, okay? But because that cleavage has happened, there is a certain tendency for the S1 subunit to fall off, okay? In which case, you can no longer bind receptor, okay? So if that cleavage is like too good, then you may have viruses that are not as efficient at binding
Yes,
receptors.
right.
So there's an optimum cleavage.
Where was this? Where was that
efficiency.
Experiment, Kathy? I missed it. Where they must use the flu H5 cleavage.
Well,
site,
they're
right?
comparing different furin cleavage sites that are cleaved with different efficiencies. If you go back to those Western blots,
Okay. Yeah.
okay, the flu guy is cleaved very efficiently.
Right. That's correct.
The wild type SARS-CoV-2 is intermediate, and some of the mono CS is even less than that. And then of course, SARS-CoV is not cleaved at all. So you can compare all of those and get those data.
Okay. So the ones that are really well cleaved, they're not as good because of the problem with S1 falling off.
we think.
That's what we think. Yeah.
Yeah, there's one other thing they mention as a possibility for why it's, it might be a trade-off with the stability of the spike. They actually say, or maybe further optimization of this site could result in higher transmissibility.
Right.
And that's when they start talking about, there's the B117 variant with mutations near the S1, S2
Right.
cleavage site.
That's right. Okay. The last experiment is the transmission experiment, and they use ferrets because not only can they be infected by SARS-CoV-2, they're related to mink, right? So,
Which I didn't know till I read that. Oh, okay.
and so not only can they be infected and they reproduce the virus very well in the nasal tissues, but they can transmit to co-housed animals. So they infect intranasally with either the furin cleavage site deletion mutant or wild type virus. And after a day, then they add in some naive ferrets, the ones that they're hoping to be infected by contact or whatever, put them in the same cage, and then they check them all daily, twice daily, or daily by nasal wash for 2 weeks, and they measure virus shedding, which they say not only by PCR, but by TCID50. So yay.
Good.
And this is SARS-CoV-2, so this is BSL-3. So wild-type infected animals have shed more virus than the animals infected with the furin deletion virus. In the wild-type group, 2 of the 4 contact ferrets become infected, so that means transmission has occurred. No transmissions from animals that were infected with the furin cleavage site deletion. No transmission versus 2. Now, I really wish they could do more. I understand these are expensive, but 2 versus 0 is kind of
Yeah.
right. And I don't know what it means. I mean, you certainly cannot extrapolate to people because, well, first of all, they're animals and it's so few. Anyway, that's the result.
This is where you use phrases like "is consistent with."
That's right. That's right.
Okay. So to me, the fundamental question here is, okay, we've got all this stuff in cell culture that says that there could be a selective advantage in the replication and or transmission of this virus if it has a furin cleavage site. So let's do an animal experiment. And the best they can do is an experiment they can do is the ferret experiment, even though they're restricted to small numbers, and they get a result that is consistent with the notion that if you have the furin cleavage site, you're more transmissible.
Yeah.
But we don't have big numbers of animals to demonstrate significant statistical significance.
All right. So then they do a competition study. They do their 30-70 wild type furin cleavage deletion site ratio. Same thing. They infect, and a day later they co-house. They measure shedding, and they look at which virus becomes dominant. And they say which genotype becomes dominant is random. In 2 animals, the wild type became dominant, and they shed a lot of virus. But in the other 2, the deletion mutant became dominant and outcompeted wild type. Isn't that interesting?
Yeah.
Just random.
They use the word stochastic, right?
Yep.
So it's kind of like it's a coin toss. Who gets the head start?
In this case, transmission was only in 1 contact of a animal that had been shedding wild type. So again, it's even less, right, than before, and only with wild type. So none of the deletion mutant infected animals were able to transmit. So they say these results strongly suggest that the furin cleavage site of the spike is a determinant of transmission in the ferret model. So very circumspect, right? Suggest and ferret model. That's about as good as you can do, right?
Mm-hmm.
So then the last thing they do is say, okay, let's look in people. Do you ever lose the furin cleavage site? So they look through they look at sequences of clinical specimens, and if they look at GISAID, which is the database that we talked about a while ago of these sequences, they could only find 2 sequences out of 100,000 with deletions of the furin cleavage site. So they did their own sequencing. They had some samples from post-mortem cases, and there they had samples from different tissues besides the respiratory tract. And they found very low levels of RNA with different cleavage site deletions, less than 1% of the whole population. In 2 patients from heart and spleen tissue. And they say the deletions are similar to what you get in vero cells when you passage SARS-CoV-2. It loses the furin cleavage site because you don't need it. You can get these in vero cells, they get in through the endosome. So they say, you get deletions rising at a low rate. And I think it's because these viruses are maybe reproducing in those other tissues where furin is not an advantage. A furin cleavage site is not an advantage, right? It's kind of like kidney cells. And maybe that's why they lost, or maybe it's just stochastic. I don't know. But I think that's pretty good evidence that you don't lose the site. And probably it's important in people, right? Maybe for transmission.
Mm-hmm.
SARS-1 doesn't have this furin cleavage site. Wasn't a great transmitter. Maybe that's it.
Right. Yeah. It kind of threw me at the very beginning of this paper because in the abstract, they say that SARS-CoV-2 has this polybasic insertion at the S1, S2 cleavage site. And I immediately said, insertion relative to what?
Yeah.
And the answer is it's relative to some other of the group 2B coronaviruses. But really, they should just say it has a polybasic sequence at the S1, S2 cleavage site instead of calling it an insertion. So
I think I'm thinking of RATG13, right? The close ancestor.
That did not have a furin cleavage site. Is that correct?
I'm looking at the picture here. Yeah, it doesn't have a furin cleavage site. So I think, Kathy, that's what they probably, because they both have a common ancestor, right? So they're saying that.
Well, I don't know. If they're going to say, it has an insertion, to me, I want to know relative to what.
I agree. So the furin site facilitates entry at the cell surface, okay, in some way. For the endosome, it doesn't matter. But that allows the virus to avoid the endosome where the IFITMs are. And I like that idea because that is a functional reason why this furin cleavage site is advantageous. So maybe IFITMs are really restrictive for coronas, and they would not be able to spread if they were getting in via the endosome. So if you don't have a furin cleavage site, you've got to go through the endosome, and there you're inhibited by IFITMs. So I think it's a nice idea. I like that very much. You can imagine that somewhere, at some point, an ancestor acquired this furin site, and that gave it an ability to reproduce efficiently in respiratory tract cells. So, and remember, SARS-CoV-1 doesn't have it, so it's very attractive, I think. So they say and the lack of mutants in humans is evidence that the cleavage site is essential for sustained transmission of SARS-CoV-2 in humans. I think that is correct. I think it is evidence that this is part of the reason why it can transmit very well. But and just to say to people who say that's evidence that it was human engineered, how would you know that the furin cleavage site would do this? You have no idea. We're just learning from these experiments, so that makes no sense. So the last thing I wanted to touch on, presence of a furin cleavage site is not uncommon. Half of human seasonal coronas, as well as MERS, have furin cleavage sites, whereas the others do not. So I just take back what I said. Furin-mediated cleavage of spike is not an absolute requirement for efficient human respiratory transmission. Okay, it's not, but for SARS-CoV-2, it might be, right?
Yeah, it's just one thing.
Yeah, it's just one thing. They make the following statement, which I highlighted in a different color. Monitoring animal coronaviruses will probably be important in predicting and preventing future pandemics. Mm-hmm. You think?
Yeah.
Okay, here's the suboptimal. We also note the SARS-CoV-2 cleavage site remains suboptimal for furin cleavage. It's unclear if this is a trade-off with stability of the spike, as you mentioned, or whether further optimization could result in higher transmissibility. And then they say, as Kathy noted, the B117 variant has changes proximal to the furin site, which are predicted to enhance furin cleavage. And I bet they're doing those experiments to see if it makes a difference in ferrets or not.
Mm-hmm. Watch this space.
Watch this space. I really like the IFIT connection. The idea is that if you get in at the cell surface, you avoid this inhibitor. I like that. It's very appealing in an anthropomorphic way, unfortunately. It's how we operate.
So I have been distracted here with looking up Camostat because one of the implications of this is that a TMPRSS2 inhibitor, i.e. Camostat, could be an antiviral. So, and one of the things about Camostat is that in some there have been clinical trials on this drug for other conditions. So from Wikipedia, Camostat's a serine protease inhibitor. It's approved in Japan for treatment of chronic pancreatitis and post-operative reflux esophagitis. Blah, blah, blah. So at least in some contexts, it's been through clinical trials and has approval. Now, I also discover that there was published in eClinical Medicine, which is published by The Lancet, on the 22nd of April. So that's what, a week or so ago, week and a half? An article that is a clinical trial result. Phase 1 or say phase 1/2, I'm not quite sure, entitled Efficacy of TMPRSS2 Inhibitor Camostat Mesylate in Patients Hospitalized with COVID-19, a double-blind randomized control trial. Blah, 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 interpretation. This is 137 patients, randomized 2-to-1, 2-to-1, I guess, treatment to placebo. Under this protocol, the Camostat mesylate treatment was not associated with an increase in adverse effects due to hospitalization for COVID-19 and did not affect time to clinical improvement, progression to ICU admission, or mortality. Small study, no miracles.
So they link to another one in the paper and clinicaltrials.gov, Camostat to reduce progression at positive patients that are at home. And this is 389 patients, phase 2/3. So maybe, what was the patient population in the one you just cited?
137.
And these are hospitalized patients?
I would have to go back and we conducted a investigator-initiated, double-blind, randomized, placebo-controlled, multi-center trial in patients hospitalized with confirmed COVID-2 infection.
So maybe
within 48 hours of admission, participants were randomized, given the drug 3 times daily for 5 days or placebo.
Interesting. Yeah, I think it's probably too late. This one they're going to do.
Probably too late.
People at home. I don't know if that'll make a difference because they probably have milder disease. So, all right.
I gather it's oral.
Yes, I think it's oral.
Did I say oral?
Yeah.
Which would be handy.
Yep. Okay. All right. Before we go on to picks, I just wanted to, we'll skip some email for today. I just wanted to show you some postcards that I got last Friday when we finished recording. These came in from Lori, who's in Spokane, Washington. She sent 3. They all arrived at the same time. So this is, Street Art by David Zinn, which was Kathy's pick. This one is called Thinking of You, EWE. Isn't that great? And
that's great.
So she writes, things I love about TWiV number 1 of 3. Learning so much by eavesdropping on smart friends. I'm just amazed they all came at once. And this was mailed on the 19th of April, so it came on the 30th. Wow. It's 11 days, right?
Postcards are very low priority.
Yeah. But look, it should be high priority. It's so cute.
That's beautiful.
It's beautiful. Yeah.
All right, so that's number 1. Then we have number 2. This is cute. This is a fisherman, I guess. Yeah, the mouse is fishing. And
what's the Zinn caption? Can you
I have
see
to
that?
turn it over because it's upside down. Nadine and the Nautical Picnic. So Nadine is the mouse.
Right.
And Lori writes, is Dixon or Rich the fisherman? Oh, it's Dixon.
Dixon.
Things I love about TWiV number 2 of 3. The women, Kathy, Brianne, Amy, aren't token, and the men don't patronize them. I'm glad to hear
that's
that.
good. That's good to hear. Yeah. That's good to hear.
No, that's great. I really like that. And number 3, this is cute. This is adorable.
What an imagination. What
This
an,
is-
he sees these things.
Yeah.
Well, this is a hat that someone put on top of the street art, right?
No, it's an orange.
No, it's some sort of one of those.
Oh, it's one of those
barrier
Yeah,
barrels?
it's a barrier.
Yeah.
Sorry, you're right.
Yeah.
And he just drew it around it. Yeah.
Yeah.
And it's called Whatever You Say and Whatever You Do. Don't ask him about thing 1 or thing 2.
Oh, right, because it's like a Seuss hat.
Yeah.
Yeah.
It's very much like a Seuss hat.
That's right.
Yeah.
Things I love about TWiV number 3 of 3, the pics. That's how I found these amazing postcards. Isn't that great? Thank you, Lori. It's really nice of you to, I mean, kind of a thing to buy 3 postcards and then get stamps for them.
They come in packages of 20.
The postcards.
Ooh.
Yes.
And you got to buy stamps, right?
Right.
Which are different from a regular envelope, right? So,
Right.
although
Right.
these are interesting, they have different fruits and things.
They're
on
fruits.
them.
So actually, those are not postcard stamps. They're
Regular. Yeah, it says
She
USA
didn't go out and
Forever.
buy special stamps, which are cheaper for postcards.
She probably didn't have them and she didn't want to be bothered. Just want to pop it into the mailbox. Anyway, zinart.com, Ann Arbor, Michigan. 2 of them say Ann Arbor and one of them says Seattle.
Well, he gets
Is he both?
asked to go places and draw things at different places.
Because these 2 are Seattle and Ann Arbor and one is Seattle. The U is Seattle, apparently.
In Seattle. Okay.
Anyway, thank you, Lori. That's great. Very nice of you.
Yes. Yeah.
All right, let's do some picks. Rich, what do you have for us?
So I think I mentioned last time that on this trip I took recently, I stopped to see my brother. And he gave me a sourdough starter that was given to him by his daughter, which I brought home. And I have never done anything like this before. So I've now baked 2 loaves of sourdough bread and a batch of cinnamon rolls. And, being a microbiologist, the sourdough starter fascinates me. Okay, because you feed this thing and it bubbles up and it froths up and then it crashes and then you feed it again and you wonder what the heck is going on. And of course, it was not started with any sort of a package thing. My niece just, mixed up some flour and water and let it sit out. And there are bugs in the universe that populate this thing and grow up. So I'm wondering to myself, actually, I'm thinking, somebody has characterized these. And sure enough, I did a little search and I found a paper called Diversity and Function of Sourdough Starter Microbiomes that's published in eLife. The last author's Benjamin Wolfe, first author's Elizabeth Landis, and they're from a bunch of different places, Tufts, University of Colorado, NC State, North Carolina Museum of Natural Sciences. I couldn't figure out exactly why that was. And there's a museum in Denmark as well. Yes. Natural History Museum in Copenhagen. And these guys took 500 sourdough starters from all around the world and did a microbiome analysis by sequencing on them. And it's fascinating. And as you might imagine, the big player is Saccharomyces cerevisiae. There are 2 varieties of this that we're commonly familiar with. One is baker's yeast and the other is brewer's yeast, but that shows up quite predominantly. And then there are 2 different bacteria that show up. There's a lactic acid bacteria and an acetic acid bacteria. I know less about these. And of course, there's all kinds of other stuff as well. But those are the really common ones. And they, report on this and talk about their properties relative to sourdough and et cetera. So I thought that was just an interesting paper.
It's really cool too, because they measure, they show you the map of where all their cultures came from in citizen science thing. And then they have data on like if you stored it in the fridge or room temperature and how often you fed the starter and how old it was, a couple going back over 200 years, whether they came from a business or an individual. They really made a paper out of it.
Yeah. And one of the sort of, shall we say, myths at this point about sourdough starters is that there's geographic variability. Think San Francisco sourdough bread, right? Mm-hmm. As if the starter that's used in San Francisco sourdough is some sort of geographic, some sort of regional specialty. But what they find, in fact, is that there's no pattern of geographic sort of specialization of these starters, that it's everything is all over the place.
This is so funny. It's so funny.
One of the authors is, we get it right here, Robert Dunn. Mm-hmm. Get this right. I'm going to mess this up. I put in a link to a subdomain of his laboratory. He's at NC State. And I get the sense he's a driver of this, and has a site in his lab that's a public science lab that is basically recruiting in a citizen science way people to participate in projects like this, and also teach them about how to make their own starter and how it works and what it's all about. So I put in a link to that as well. He's got a site called Wild Sourdough. Capture wild microbes and turn them into bread for science.
That's great.
So you can have a look at that.
I have to do this on TWiV as a snippet. This is great.
I thought that paper might be good for TWiV. Yeah.
Yes. We did one similar to. They do similar studies with wine, right? You pick. Mm-hmm. You go to certain grapes and you ask what's on these grapes, because the natural microbes on the grapes contribute, right, to the fermentation, and try and figure out what makes the best wine and so forth.
Well, the thing that gets me thinking is, we're such in the 21st century here, we have a tendency to be germophobic, especially with a pandemic going around. And the thought of taking a mixture of flour and water and just setting it out on the counter for a few days until it bubbles up.
Yep,
okay.
Yep.
And it obviously has all sorts of junk growing in it, is, I think would be horrifying to lots of people, but that's fundamental to a lot of our food stuff, yogurt, cheese, beer. I had a friend who, in the College of Medicine, you always introduce your seminars by talking about your pathogen and what terrible disease it causes and how your basic science is going to cure that disease. And this guy worked on yeast. And he would get up in the beginning of his seminars and say, yeast causes beer. And that got everybody's attention.
Yeah, right? That's great. Hey, it's Louis Pasteur. He's the one who started all this.
Yeah, when you're talking about what you find in your kitchen, it reminds me of I sent that New York Times thing about how the Pfizer vaccine is made and how stunningly clean everything is. And I said to somebody, for anybody who's worried about putting toxins into their body, like from a vaccine, should look at this and see how much cleaner it is than any home or restaurant kitchen.
Yeah, really good point.
Very cool. Yeah. Love it. Kathy, what do you have for us?
Well, I have two astronomy pictures of the day. One is from today, and it's so cool. It's the International Space Station with the sun in the background. So there's sunspots and the ISS. And then they've blown up the ISS, and you can see Crew Dragon attached to it.
Yeah.
Oh, wow.
Cool.
It's just an amazing photo. And then the other was one that I saw last week sometime. And And it's this doubly warped world of binary black holes. And I watched the video and I read the caption, and I watched the video and I read the caption, and I can't really tell you that I understand it, but it's really kind of fascinating to watch. So binary black holes in this video reconstruction.
Neat.
So check it out.
Beautiful. I love the ISS picture.
Yeah.
It's gorgeous.
Yeah.
So
Wow.
are these black holes really far away from each other, or are they together? That's why they're binary, I guess, right?
I
A pair.
I don't
It
even
says
remember that.
A pair
Of orbiting
Yeah.
supermassive
Yeah.
black holes.
Wow.
Yeah.
That's weird. I would think they would like go into each other or something.
Yeah.
Little do I understand black holes. Okay.
Yeah. Yeah.
That's pretty cool. My pick is a video by our musical friends in Santa Fe, Gina Browning and Joe Illick, called Mr. Labman. Now, those of you who listen to TWiV may remember a couple of months ago, Dagfinn Rosnes sent us lyrics to a COVID song, which was meant to be sung to the tune of Mr. Sandman. And I remember saying, oh, it would be great if someone sang this, and they did.
Mm-hmm.
Gina sings it, Joe plays the piano, and they have great props.
They do.
They have great
Yeah.
props.
Yeah.
And once again, they've got principles of virology sitting on the piano.
They got a
They
face
wind up
mask.
taking drinks out of 50 ml
I love
conical
that.
tubes
I
or
love
something
that.
equivalent.
And
Yeah,
then
that's
at
great.
the end, no vaccine was harmed in the making.
Yeah.
It's just great. Thank you, Gina and Joe.
I wonder if they've been in touch with Dagfinn.
Well, Dagfinn, are you listening? And
I hope so.
tell us what you think. And they loved your lyrics, and they're very cool.
Yeah.
So, I mean, this is very well produced. This wasn't just taken on your iPhone, folks. It's well lit and it's well recorded, and
Oh, yeah.
it's
Well,
edited.
they're professionals.
They're professionals, yeah.
Mm-hmm.
So, thank you. That's very cool. We have
Yes,
a
thank
listener
you.
pick from Joy. Abandoned America, spooky, beautiful, thought-provoking look at America's abandoned buildings. Part art, part history, part expose of American wastefulness. Somehow I think this is one Kathy would like. There's a website, abandonedamerica.us, with lots of great photographs.
Yeah. So
It's weird.
photos of Detroit like this are well known. And in fact, I was able to find a book and another site with photos of Detroit. But these are the only thing I think this site lacks is a search function. I wanted to search it for locations or things like that, but it's got some pretty cool photos.
And they're amazing pictures.
Yeah.
They have an abandoned Georgia. Might like that. Might find your old home there. Who knows?
Yeah. Yeah. Yeah.
Thank you, Joy. And that'll do it for TWiV 751. Show notes are at microbe.tv/TWiV. You can send us questions or comments, twiv@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back in a few days. Another TWiV is viral.